It's absolute pleasure to be in Sweden, to be in Göteborg, and be invited to join this seminar. And obviously tomorrow, as Anna and Anna mentioned, we have Leo's defense of his thesis, which is always a very nerve-wracking event, and we're not going to preempt anything. Although we all know the merit and the quality of this team and work. And it's a great honor to present in front of you and people in the audience. Actually, sort of there is some connection between, obviously there is a lot of collaboration that we did in the past with KTH, but I'm also a little bit KTH alumni. So when I did my PhD, I spent 6 months at Yakemsky Institute in so it's an Ostra Station, and then sort of with the KTH. And as you pass the KTH, there was a Yatkemsky Institute building, and I was working for 6 months with Professor Per Kleisen. Oh. So we are all kind of it's a very tight network. So we never go really far. So it is really pleasure to be here and to an extent reconnect with my Swedish past. So it was a fantastic time. So out of my PhD, I spent some time in industry in England, working for Unilever, and then I had an opportunity to join the University of Queensland in Australia, where I was part of the Australian Research Council Centre of Excellence in Plant Cell Walls. And this is where my sort of expertise in plant cell walls and polysaccharide assembly really kicked off, where we were really trying to understand the xylem biosynthesis. And I'm going to talk a little bit about that. So, and then I had an opportunity to join the University of Nottingham. And last year, I've joined the University of Leeds, which is a northern part of the United Kingdom of England. What we do is most of the our work is really in this multi-scale domain. So we are exploring the rheological and micromechanical properties of polysaccharides and polysaccharide assemblies, from the nanoscale to the macro, and from macro to the nanoscale. So sometimes some projects we start looking at the plant cell walls, and we try to understand what are the molecular interactions that govern this assembly. The other project, we start with molecules and we try to build the structures. And I think this is very much what FibRe Consortium is also very much leading this area. So as you know, we're all very interested in cereals. They happen to be the staple for carbohydrate and polysaccharide research. We are also interested in cereals. We are also interested in polysaccharides from the vegetables and fruit, in particular sugar beet pectin and okra pectin, so on the pectin side. But we are partial to our little plant. It's a family of plants called the plantago. It's a family of plants. They grow everywhere, and we'll talk a little bit more about it. And really, most of the research that we've been doing in the last probably 10 years was really going deep into understanding the structure of these unique polysaccharides. As always, these days, it became a fashion to start the acknowledging at the start. So in case we're running out of time, there is a lot of, and obviously, since we are in this consortium, I would like to acknowledge my collaborators at KTH. And I'll show a slide to remind everyone about our collaboration. So in my group, we have like this 4 different perspectives. One is the fascination. So this is where you look at the material and say, wow, this is amazing. The other one is more kind of vision. So we want to remove all the petroleum-based plastic. We want to replace it with the bio-based materials. We know that plant cell walls are not disintegrating in water, right? We know that. We know that paper, if you put it in water, it disintegrates. But plant cell walls do not disintegrate. So understanding the reason why this is happening, and we know why this is happening, but how we can manipulate that, that is the biggest problem. And finally, solving problems. Sometimes we collaborate a lot with industry, and solving the problems becomes an important part of our group's work. And finally, developing tools. We are not only users of some tools, we also try to develop new tools. So this is one of the new equipment that we've established, and this is the combination of super resolution confocal with fluidic force microscope. So that enables us to do the AFM with the super resolution microscopy, and or for example, using the fluid FM, you can manipulate the cells, or you can selectively hydrolyze certain area within the plant cell wall. So there is a lot of work that has been done now in the bacterial space. They're really sort of, there's a lot of use at the moment in the characterizing biofilms. So this equipment is available so that you know what it can do. It has a very open architecture. You can put the whole plant underneath. So it's not like a typical AFM where you kind of, oh, you have to do everything. I can show I have a separate deck of slides about this machine. So if anyone is interested, I would be more than happy to share these slides with you as well. So this was the launch event, which was happened last year, 27th of November. And we really were looking at creating the community. So if there is an interest in doing this type of work or discussing, please come and discuss with me. Another area of development that Leeds is particularly trailblazing is alternative protein. So Anwesha Sarkar, my colleague, she was leading a bid together with our colleagues in the University of Sheffield, the James Hutton Institute in Dundee, and University of Leeds, obviously, as a leading partner, and Imperial College in London, really looking at creating a new ecosystem for the alternative proteins. As we know, alternative proteins is something that is going to happen, but at the moment is a little bit like at the stage of EV vehicles 20 years ago. Everyone knows that EV vehicles will be the future, but the batteries were not good enough. The other things were not good enough. And I think that's what happens with the alternative proteins. So this is a big project. Is a 37 million split between the government funding and the industrial contribution, with the government funding more than 20 million. So it is really looking at the economic, environmental, health, and societal impacts, and it's really should operate. At the moment, there is a lot of fundamental work and translational work happening, but we believe in 7 years that will become a standalone center with its own ecosystem, and there will be a continuation and of that line of work. Now, without further ado, I will start my scientific talk, since we here to exchange our scientific ideas, and that's what I wanted to basically introduce you to, is just how we went thinking about different problems related to polysaccharides. So the role of molecular conformation and hydration shell water in the assembly of complex polysaccharide. That's actually the topic. The sweet battle is a clickbait, right? As you know, on YouTube, we'll put this. Anna told me that we'll put it on YouTube, so we were scared. But now we will go with the flow. I mean, if it's on YouTube, let it be. So all my career, I've been working on different systems, but it's been always comb-like polymers. So whether these were synthetic combs, or that was mucins, which is the proteins glycosylated, or polysaccharide combs, that was really my passion. I want to understand what is different about combs that linear polymers don't have. And we know that the combs and the glycostructures are very important. I mean, they're very important in glycoprotein. This is the COVID spike protein. And in green and magenta, you can see glycosylation on the protein. And you can see it's all covered with sugar. So when we think about the importance of the protein binding site, they're actually very important for the recognition and for the drug delivery. But we know that in reality, it is the carbohydrate that do a lot of work that mediate the binding of those proteins. And the reason why no one is looking into people look into this, but why there is always less effort is because it's very difficult to characterize polysaccharide. So everyone goes, okay, probably it's more important, but we can't do this. So we will do things that's less important, but we can actually have a meaningful result within a single PhD. And therefore, anyone who's doing PhD in the carbohydrate, they need to be very proud because they're trailblazing a very complex field. So mucin, I already mentioned, that's another sort of glycoprotein that has a high abundance of sugars. Obviously, peptidoglycans, where you have a polysaccharide chain, but then proteins, oligopeptides that are cross-linked. And finally, our favorite polysaccharides that we're going to talk about today. But even DNA and RNA, you can think about it as a fancy pants sugar with some very fancy pants side chains. And it happened to be these fancy pants side chains that overshadow the presence of sugar. But actually, if you think about it, at the very early stages of evolution, I don't think people were kind of the molecular soup was choosing those things in a very rational way. They just happened to settle on this configuration. But this sugar motif of sugars and phosphates, I think that becomes very universal across all biological species, and I think that's what is fundamental. And what the side chains give, they give this specificity, but it is an add-on to the original structure, which is essentially a polysaccharide at heart. So why we're interested in sugar, this is the slide that I'm taking from my mentor, Professor Tony Caulfield, and it's really about the complexity of sugars. So when I have lectures about sugars for especially first-year students, I'm showing them this model of a glucose molecule, and you just flip between the two chair conformations, and you say, look, guys, it's two chair conformation, but one is where the equatorial, the OH groups, and the other one where they're all sort of sticking out, right? And this is like magic. You just twist it, and you can see that the structure is completely different. And because biology communicates with this touch, they see and they feel the shape. And that's why the different complexity of the sugars contribute to this what we call the sugar code that communicates the information. But it's very hard for us to understand, and we decipher this code. And even more difficult is actually to learn how to write this code. So this would be an ideal case scenario. We'll create a polysaccharide that would have a specific function. Unfortunately, it was long realized in the DNA. They can do DNA origami and things like that. They have a lot of control. But this is something that we definitely will get at some point. But we always should work towards really this fundamental understanding of how polysaccharides interact and encode information through their very complex structure. So why is it important? Because we use packaging, and I don't have to introduce you to packaging in this audience. This is this is the leading group in that space. Obviously, foods, biomaterials, and drug delivery. That is our collaboration with Francisco and Jenny Berglund a few years back, where we really looked at the bacterial cellulose networks that were combined with the hemicellulose from spruce and birch, and we were trying to create those materials with the unique extensibility and unique mechanical properties. And our contribution to this work was really providing the framework for the analysis of the mechanical properties and understanding the effects of the poroelasticity associated with those materials. So of course, we are all interested in xylans, Arabina xylans in particular. Tomorrow we will have a Leeds defense. So I thought I will start with Arabina xylans. And that's how we started when we were part of the Australian Research Council Centre of Excellence in Plant Cell Walls. We wanted to understand the xylan biosynthesis. There are a lot of xylan synthases that haven't been quite crystallized at the time. People didn't know, and we couldn't get enough material. We couldn't afford to go through all the linkage analysis because we want to do these. So we need a lot of material. And therefore, someone suggested, oh, I actually know the material where there is a lot of xylan. And they've introduced us to Plantago. They said, actually, psyllium, all this mucilage, and there is a tons of it, it's almost all xylan. And we were kind of like looking at it, okay, can it be our Arabidopsis of the plant kingdom? Can it teach us anything about the xylan biosynthesis? And what is very important, we started looking at the different mucilages of these materials, of these seeds that produce this mucilage. And we realized that they have all the same template, xylan template, but the structure is very different because there is a different way of decorating these xylan backbone. So different decoration resulted in the different properties. So each species of Plantago was adapting to a specific environment in terms of drought, in terms of drought resistance, how dry it is, how wet it is. What does it remind you? What does it remind you? The same template, but then you adapt it to different environment. Of course, it reminded us Darwin finches because that's how the evolution works. So you have the template, it is successful, and so you have this opportunity to tap into the different Plantago species, different types of psyllium, to actually understand the structure-function relationship. So we started working, and you have the seeds. Seeds are coated with a husk, and a husk, if you put it in water, produces a gloopy substance, a jelly-like mucilage. So at that time, a very talented PhD student, Long, came around, and we were trying to develop a very simple microscopy tool for phenotyping Plantago mucilages of different seeds. So we in Adelaide, they had a collection of the irradiated seeds with the different mutations, and we wanted to phenotype them. And we started looking at the staining. So we came up with this staining with the direct red 23 and the chalcofluor white. And then it was like one of those moments, why there are 2 layers there, or actually 3 layers. And we started, oh, that's odd. Why there is a difference? So there is a different chemistry. And I said, Long, I think it should be a quick paper. Let's just extract different. That's what I said. I admit I was wrong, but I said that. It's going to be a quick paper. We're going to show that there is different chemistry that leads to different structures and things like that. It's an amazing way how I'll try to see if I can actually show you. So I don't know if you can see really well. So this is the first layer. It contains some pectin, but then you have 2 differential layers that are stained differently. And we were trying to extract different materials from different layers. And what we found is this viscoelastic layer from the outer side, which acts as a little bit as a propellant to start the mucilage expansion. Contains conjugates of Arabina xylan with pectin, and it forms a viscoelastic fluid. There are a lot of interesting things about it, but let's forget about this. What we are interested in these 2 fractions. One is the fraction that melts at about 40 degrees, and it's from the inner part closer to the seed. and it's arabinoxylan. It's heteroxylan essentially, and it is a weak gel. It melts, and the other fraction forms an elastic gel that is thermostable. Even if you put it in the autoclave, it wouldn't fully melt. It will still retain the gel property. So totally different behavior. And yet we thought, okay, fantastic. We've analyzed arabinose to xylose ratio. It is the same. It is arabinoxylan. And I say, okay, that's going to be a little bit longer paper long, but because it seems to be that the linkage composition is different. We're going to show that linkage composition is different, and then the property is different. And then we do the linkage composition, and then we say, all right, they're the same. So the linkage composition is the same, molecular weight is the same, and what we're finding is that if we do things carefully, we actually realize that the level of the backbone decoration is almost more than 90%, is more than 95%. Conservatively 90%, but it's actually more than 95%. So this is almost every single backbone sugar has a side chain. And that's what makes them unique. So they have very similar linkage composition, similar molecular weight, but distinct gel properties. And that was the start of our quest. So we wanted to understand what drives these difference. And then this quick paper turned out not quite quick, and then there were a few papers to follow. So we want to understand what is the mechanism of gelation and why they're different. So instantly what we've realized is that none of these gels are gels. So they're not true gels, they're physical gels, because you can see this dependency of the G prime and G double prime, these moduli on frequency. As soon as you see them sloping, not really flat, you know this is not a true gel. This is a physical gel. That means that there is a certain rearrangement. There is a breakage of the hydrogen bond or any other bond, and there is a reformation one. So we did all the characterization looking at the hydrogen bond interactions. We looked at this large amplitude oscillatory shear rheology, trying to understand the behavior of these materials under large deformations. Then we looked at the dissociative states using the time temperature superposition approaches, as well as the newly developed the Arrhenius approach that we kind of adapted and modified to extract the numbers. And essentially what we found is that these two xylans are different in a way how they have these junction zones. So the one that melts has one type of junction zone that dissociates very quickly, and the one that is only soluble in the strong alkaline solution is actually has the junction zone that are much higher in energy. It has also this weak junction zone as well. So it has two types of them. And that's why there is a lot of interesting behavior comes, because there are two types of the junction zones. But it has a very unique way how it is structuring, because the relaxation profile and the therefore rheological properties are totally different. Totally different energies. And you can see that. So we have this determined the energy of the weak hydrogen bond junction, about 100 kilojoules per mole, and the strong hydrogen bond junction at 450 kilojoules per mole. And that's what drives this behavior, where they can fuse together. You can have this rearrangement of molecular structure, but still, because of very strong energy of this junction zone, they can be rearranged, but they are not dissociating under normal conditions in water or higher temperature. You need alkaline solution to completely disrupt, because it's so high energy. So then we started to look, is there any difference in network structure? So we did the SEM, we did the SANS, and with SANS it was very interesting. Then we realized that the melting temperature is changing. So obviously, and then people were saying, oh, yes, the melting temperature, yeah, if you move to D2O, melting temperature, always the changes for gels. Sometimes it's 5 degrees and things like that. For these guys, it was 40 degrees change in the melting temperature. Absolutely huge change in the property once you go between H2O into the detail. So we started looking at the secondary structure. We find some small differences in the secondary structure, but we look at these twofold and threefold screw motifs in those xylans, and we try to identify the differences. And what we identify is that our easily melting fraction is mainly twofold motif, and threefold screw is mainly our alkaline fraction. So this is very interesting. So the twofold motif, which is much tighter, seems to be a stronger gel compared to a threefold screw motif. However, this is just an observation. This is still not an explanation of what's going on. You can see the different structure using the AFM, for example. You see a different degree of coiling, that the twofold screw motif in the alkaline fraction leads to a much tighter and much more coiled structure compared to a more open threefold screw motifs in the water-soluble fraction. And then we looked at the apparent diffusion coefficient during the spin-aqua NMR and so MRI, and then we find a quite significant difference in the T2 relaxation time. And we thought that that means that the water is structured different around those xylans. And so we took the inspiration of the of solvent-mediating interactions, so like these micelles, surfactant micelles. So the surfactant micelles, they don't want to come together because obviously that reduces entropy. And actually, people say, oh, there is a hydrophobic things, they like each other. Actually, hydrophobic things don't like each other, really. What happens is that water that is surrounding these hydrophobic things really hates being around these hydrophobic tails. So when those hydrophobic tails assemble in a micelle and they form this core shell structure, a lot of water gets liberated and it gains a lot of entropy, and as a result it stabilizes that structure. So yes, there is some short-range hydrophobic interaction, but the main driver for the micelle formation is the entropic contribution from water. So we thought about a similar thing in this instance, and in this instance we thought that, oh, there is a water that is bound to these molecules. And obviously, this water is in an entropically unhappy state. And what happens is that if they start gelling, they share some of the water that is in the absolutely dire state, but then another water essentially gets liberated. And that was like an interesting idea. So you have low entropy water, then there is a drive to assemble and form a gel, form a junction zone, with some water present still in the junction zone, otherwise they can't slide and rearrange. This water is extremely trapped, so it entropy even lower. But then, because you liberate some of it, you get some gain. So, and therefore you go from something that is unhappy to something that is relatively neutral. So we wanted to prove this hypothesis. And we looked at the ochre pectin. So we extracted another polysaccharide from ochre. And in this case, we realized what we needed is this super high molecular weight. So this is in the tens of millions molecular weight. And that's why it has this super elasticity. And what we've done is that we wanted to change the solvent property because we now want to modify our hydrogen bond environment. And we actually introduced the glycerol into the system to look how the change in the solvent leads to the different relaxation time, having in the hypothesis about this happy, unhappy interaction with water. So what we found is that you have this flexible transition between the flexible chains in water and the low concentration of glycerol to the rigid chains. Rigid chains as the higher concentration of glycerol. When you go to 60 and 80% glycerol, you start seeing this rigid chain formation. And the reason why this is happening is because there was a lot of self-assembly. So essentially, you start with something that is flexible, then you go to a semi-flexible state, and then it goes into a rigid state. And so this is my postdoc, Palab, who is now a fantastic fellow at the Technical University of Munich. And he did this fantastic modeling we did for this random galacto-Uranus chains to understand its conformational changes as a function of the solvent quality. So in this case, we use the explicit solvent. So we put the glycerol in water and we looked at the radius of duration and compare it with experimental data. And so you can see now there is a difference in how in the affinity of this change. So as you increase the water glycerol ratio, essentially the chain becomes collapsing. The chain becomes more rigid. It collapses on itself, which gives its rigidity, and as a result, we observe these effects associated with the manifestation in the rheology of these polymers. So what we found, I'm 5 minutes. What we found is that the reason why they're behaving like that is because the glycerol now is getting underrepresented in the first, second, and the third hydration layer. So essentially, as you increase the glycerol concentration, you assume that the glycerol concentration should be actually the same in all hydration layers. But what we observe is that the glycerol concentration is overrepresented in the fourth hydration layer, but it is heavily underrepresented in the hydration layer closer to the polymer chain. And that phase separation then brought us a different idea. So the maths doesn't work with this idea of the happy and unhappy water molecule, because in order for this maths to work, you need a very, very specific energies to say, oh, as soon as you change something, it should stop working, because you break the balance between the entropy of those super confined, and let's say liberated water molecules. So it is a model, but it is a kind of model that is not operational. It's not a generalizer. You cannot generalize this model. It's very specific in terms of the balance of different energies. So we looked also at probing this overrepresentation, and we looked at the zeta potential measurements, which is a very simple measurement as a function of the Debye length. And essentially what we were probing is that sodium chloride was migrating inside that water-rich layer, and therefore the zeta potential was changing because of this preferential migration of the sodium chloride, which gives us an evidence, experimental evidence, of this enrichment of the hydration shell water around the polymer compared to the bulk concentration. And we currently, Palau has collected fantastic X-ray scattering data that confirming these hypotheses as well. So based on these ideas, we developed a new model of how things are working. And things are much more subtle, but they are universal. So what we are thinking about is that because of this difference in the mobility and diffusivity of water, essentially you have this gradient of density of water as you go away from the polymer chain. So the density of water is not the same because there is a lot of side chains. And because there is the gradient of the density, then in order to capture all this energy of the system, you actually need to take this into account. This is a non-linear effect associated with the non-homogeneous density of water around the chain. And then you can bring the density functional of the polymer solution in the form of the Cahn-Hillet equation that enables you to separate the square root of gradient terms from the zero term. And as a result, essentially, if you come to two molecules come together, they reduce essentially the surface area of this gradient. And as a result, they minimize this nonlinear contribution to the free energy. And this essentially absolutely universal mechanism. If you think about the polyethylene oxide it also has this gradient of the density, but the gradient of the density is 0.000000. You never can measure it. It doesn't matter. It doesn't matter at all. At all. But when you have the side chains, they break the symmetry. And that was the critical moment why it's so potent in those branched polysaccharides. This is because the side chains, they break the symmetry. You can't just average around because you have a polymer chain and you have a cylinder. Because you have the side chain, you start breaking this symmetry. And this broken symmetry leads to that square gradient terms being non-zero. And this is kind of important because then we can start thinking about how we can create the side chains and their distribution, that they have a controlled way how they break the symmetry of the molecule. And on that, I will thank you for your attention.